All right, welcome to our last class this semester. I want to review the four main biomolecules. So here is an infographic, and you have four main biomolecules: carbohydrates, lipids, proteins, and nucleic acid. High energy compounds, it is considered important in a molecule in our bodies, but the main ones are carbohydrates, lipids, proteins, and nucleic acids. Here is a brief overview. We have carbohydrates, which are composed of polysaccharides, disaccharides, and monosaccharides. We have lipids, which include triglycerides, which are made of fatty acids and glycerol. We have proteins, which are composed of peptides, and amino acids make up those peptides. We have nucleic acids. That breaks into two parts, RNA and DNA, and these are composed of nucleotides. We're going to go into detail about each one of those, but this just kind of gives you a breakdown, a nice little overview. So the first is carbohydrates. Now, carbohydrates, you might see written like this, where N is the number of carbons in the molecule. You have two types of carbohydrates. You have the simple carbohydrates and you have the complex carbohydrates. There are two types of simple carbohydrates: monosaccharides. Mono means one. So these are simple sugars that include glucose, which is your blood sugar, galactose, a type of milk sugar, and fructose, which is a sugar found in fruit. They all have the same chemical formula, C6H12O6, but their structures are different. And if your structures are different, you know you behave differently. You have different properties. The second type of simple carbohydrate is a disaccharide. Di means two. So disaccharides have two monosaccharides together, and they form during a dehydration reaction. So you're losing a water molecule from your monosaccharides, from two monosaccharides joining together. These disaccharides include lactose, maltose, and sucrose. Sucrose is your table sugar, all linked together by glycidic bonds. Now, the second carbohydrate is the complex carbohydrates. So these, you have polysaccharides, which are long chains of monosaccharides that are linked together by glycosidic bonds. And this includes glycogen. Glycogen is your glucose form or storage. Glycogen is your glucose storage form, and in our bodies and in other vertebrates. Glycogen's broken down into glucose whenever our body needs it, when our glucose levels decrease. Now, fatty acids are long-chain carboxylic acids, and they're derived from animal fats, vegetable oils, or phospholipids in biological membranes. There are more than 500 that have been isolated from various cells and tissues, and most have between 12 and 20 carbons in an unbranched chain. This is unbranched. You can see it's just a straight chain. That's what the fatty acids look like. Now, triglycerides are an ester. Remember the ester? You have the oxygen that combines two alkyl groups. Triglycerides are an ester that consists of three fatty acids and a glycerol. So here's your glycerol up here. And then you have, so this was your glycerol. And then you have 3 fatty acids right here. And your body creates the fatty acids during the digestion of oils and fats. So one thing I want you to remember is that fatty acids are composed of a glycerol and 3 fatty acids. That gives you your triglycerides. Now, saturated versus unsaturated fatty acids. Saturated, you have only single bonds. So here you have an example of saturated fatty acid. Only single carbon bonds throughout. Every single bond is saturated with hydrogens in this carbon molecule. That's what it means. There aren't any double bonds between carbons. These animal fats are saturated. They're typically solid at room temperature. Some plants oils can be saturated as well, like peanut oil. And saturated fats tend to have a longer shelf life than unsaturated. Unsaturated fatty acids are plant-based, and they have one or more carbon double bonds in the carbon chain. Trans fatty acids are solid at room temperature. The trans refers to the hydrogen being on opposite sides of the double bond. So here you have a trans fatty acid. This is a cis fatty acid because those hydrogens are on the same side. Those hydrogens will give a kink. That's called a kink. It's like your hose. You want your hose to be straight, but sometimes you get a kink in it and it comes like that. So if you have a cis unsaturated fatty acid, that double bond right there will cause a kink as these hydrogens are trying to get in the same area. A trans fat, sometimes you've heard of the trans fats, the hydrogens are on the opposite sides of the double bond. And so the trans fatty acids are able to just lie straight right here so that they can be packed more easily. The cis fatty acid, because of this kink, it's going to be a liquid at room temperature because here, this is like your cooking oil. Because of that kink right here, they can't stack nicely. And if they can't stack nicely, they can't make those good London dispersion forces at multiple points. And so if they can't make those London dispersion contacts, they're not going to be able to hold tightly, which means they're going to have lower melting points. So that's why they're liquid at room temperature. The saturated and the trans unsaturated fatty acids are all solids at room temperature. Now, let's talk about omega-3 fatty acids and phospholipids. Omega-3 fatty acids are required for biological processes, but our body doesn't make them. Omega-3 fatty acids are needed. They reduce inflammation and the risk of heart attacks. They lower blood pressure and inhibits blood clotting. Sources are from fatty fish like salmon, nuts, and avocados. These are great sources. unless, of course, you're allergic, and then you need to look at something else. I have a son who's allergic to salmon and nuts, so avocados are it for him. Now, phospholipids has both the hydrophobic and hydrophilic part. Remember, hydrophobic is water-fearing. Hydro means water. Phobic means fear. And then this is water-loving. Hydrophilic means water-loving. So the phospholipids, they have their hydrophilic tail and their hydrophobic, sorry, their hydrophilic base head and their hydrophobic tail. So the tails go in, and then you have your hydrophilic heads here. Now, the cell membrane consists of the two layers of phospholipids, and it acts as a semi-permeable membrane in our bodies. Steroids are another important part of our body, and they play a role in reproduction, absorption, metabolism regulation, and our brain function. Cholesterol is the most well known as our cortisol. Cortisol is activated during our fight or flight response, or flight or fight response. It's activated in times of stress. Steroids will alter electrical activity in the brain, and they're used in medicine as anesthetics. So here is cholesterol. We have heard about cholesterol. Whenever you go to the doctor, whenever you're older, they'll measure your cholesterol levels as part of your physical. You get to a certain age where they're like, all right, let's check your blood work and everything. And then this is what cortisol looks like. They're both considered a type of steroid. Proteins. So steroids, omega-3 fatty acids, saturated and unsaturated fatty acids are all part of the fatty acid category of biomolecules. Now we're looking at proteins. Now, amino acids, they all have the same basic structure. They have a carboxyl group bonded to an alpha carbon that is also bonded to both an amino group and a hydrogen. So here you have that basic structure. You have a carbonyl group that's bonded to a carbon, and they call it the alpha carbon, that's bonded to an alkyl group and an amino group. Now, there are 21 amino acids in proteins. 10 of them are essential amino acids because we don't produce these. We have to eat them and get them through our diets. So it's really important that you have a well-balanced diet to get everything that your body needs in order to work optimally. Your body is just like a car. You need the maintenance, you need the right fuels. If not, if you don't take care of it, you're going to have issues that you might not be able to reverse. So make sure you take care of it. Make sure you're giving your body the correct foods and that it needs in order to produce these essential amino acids because you need it. Now, peptides are composed of two or more amino acids, and they're held together by a peptide bond, where the amino group of one connects to the carboxyl group of the other. Because remember, you have the dipoles here. That's going to be positive. That's negative. Then you have that negative dipole and this positive pole here. Nitrogen and hydrogen form a polar bond as well, with nitrogen being positive or negative and hydrogen being positive. This hydrogen can therefore be hydrogen bonded to the carboxyl group or to an amino group on another amino acid. So that's why hydrogen bonding is so important in protein folding and in DNA double helix, because each of these amino acids hydrogen bond to form that double helix. And that doesn't look very good, but it doesn't look very much like a double helix, but you got the gist. Now, enzymes are our body's forms of catalysts, and they can both form and break peptide bonds in the body. So enzymes come are very helpful and are a big deal in our bodies when it comes time to look at DNA. Okay, let's look at protein structure. There you go. Now, the structure of protein is integral to its function. If the protein loses its structure, which is called denatured, if it loses its structure, it can lose its function. There are three different structures of the protein. You have the primary structure, which is just your amino acid sequence. You have your secondary structure, and it's its local interactions between polypeptide chain. So primary, you're dealing with just the amino acid sequence. What's there? Okay, I'm sequencing. Now you start getting those peptide bonds and interacting, forming the polypeptide chains. Your tertiary structure is the overall 3D folding by interactions between the R groups, those alkyl groups on, I'll show you right here, those R groups, those side chain groups here in your peptides. Okay, then you have your quaternary structures. And your quaternary structures, that's the orientation and the arrangement of amino acids in more than one chain. So you have primary, that's just your amino acid sequence. So that's just the basics. Then you start having the local interactions between polypeptide chains. Then you get 3D folding and the interactions between the R groups. Then you have the quaternary structure, and that's just more on orientation and arrangements of the amino acids into more than one chain. So here is a visual. Primary protein, this is just your sequence. Each one of these is the amino acid right here. So you're looking at, is this the right sequence? Okay, we've got the right sequence. Now we look at the secondary protein structure. We're looking at hydrogen bonding of the peptide backbone and causing it to fold into a repeated pattern. Then, once we've folded into the proper pattern, we have the tertiary protein structure, and we've got the 3D dimensional folding. And that's where you have the side chain interactions. Then you have the quaternary protein structure, and in this you have more than one amino acid chain. So finally, you've built up here, you have multiple tertiary protein structures. And the quaternary protein structure, you have more than one amino acid chain. Let's talk about DNA and RNA. Both are nucleic acids and regulate gene expression. And they both have a 5-carbon sugar backbone, a phosphate group, and a nitrogen base. They're similar. They have similar functions in regulating gene expression, but they have distinct differences. The DNA has the sugar chain of deoxyribose. That's where DNA comes from. Deoxyribose nucleic acid. It's found mainly in the nucleus of the cell. It has a double helix with sugar and phosphate on the outside. So the sugar and phosphate are on the outside of the double helix. This also uses hydrogen bonds to hold the nitrogenous bases together. So that nitrogen base, hydrogen bonds, in order to give us that double helix structure. DNA controls cellular activity by turning on and off genes, and it's mainly found in the nucleus of the cell. So DNA is in the nucleus of the cell primarily. And the nitrogen bases are stacked inside by pairs. Adenine pairs with thymine, and guanine pairs with cytosine. So if you had the sequence AATTGGCC, if you don't adenine pairs with thymine, the base pair would be T, T, A, A, C, C, G, G. The way I remember it, G, C, they're very similar. G pairs with C. And then by default, A pairs with T. Now, during replication, each strand is copied, and during this replication period, that is when mutations can occur, just by a transcription error. So we do have like an editing portion towards replicate during the process of replication, but it isn't always caught. So it's like whenever you are copying down some information, the majority of it's correct, but sometimes you might just say and phosphate on outside and you've skipped the. That happens sometimes whenever you're copying something down, like taking notes. This happens when DNA is being replicated as well. And if that happens, that's whenever you get a gene mutation. Like I said, we do have an editing system in our replication process, but sometimes it misses things. Let's look at RNA. We had deoxyribose nucleic acid. RNA stands for ribonucleic acid. Now, it is mostly involved in protein synthesis. So it's found throughout several places of the cell. It's in the cytoplasm, it's in the nucleus, and there are 3 different types because RNA has such an integral part in the synthesis process. So the first type is mRNA. The M stands for messenger. So this messenger RNA transmits messages from the DNA to ribosomes for protein synthesis. Then you have RNA, which is ribosomal RNA. This is a main component of ribosomes and the place where mRNA, or messenger RNA, binds. Messenger RNA is kind of like the mailman. It takes the messengers all around. So this is a mailman. You give him a letter and he transports it. So he takes it to the RNA and he docks right there in the ribosome. RNA ensures alignment of the mRNA and the transcription RNA, which is the tRNA, and the ribosomes, and catalyzes the formation of peptide bonds between amino acids during protein synthesis. So RNA is involved in protein synthesis, and that's what starts the formation of those peptide bonds. And then you have transcription DNA, or transcription RNA, excuse me, and it carries the correct amino acids to the site of protein synthesis. So once everything's all good, it carries that correct amino acid to the site of protein synthesis. Now, the nitrogen bases are stacked in side by pairs, just like they are in DNA. Adenine is again paired with thymine, but the difference is guanine is paired with uracil. So instead of being paired with cytosine, it's now paired with uracil. So if the sequence of one strand in your RNA is AATTGGUU, its pairing would be TTAA. That doesn't want to write. UUGG. So one of the bases is different in RNA. It has uracil instead of cytosine. Now, let's talk about using fats, carbs, and proteins. Our body will use all of them as a source of energy, but the first one that it will use is carbs. The carbs are first to be used to fuel our muscle and our brain. But if you don't have enough carbs, that's whenever your body moves to the fat reserves. These are used as a source of energy after the carbohydrates have been fully utilized. And fats aren't as efficient as carbs. They have a key role in hormone production and brain function. Your brain is almost 60% fat, believe it or not, and energy storage. But knowing that your brain is about 60% fat helps you realize why fatty acids are needed, especially those omega-3 fatty acids. Those are important for your brain health. Proteins are the final fuel to be used in the body, but only in the cases of starvation. So if you are literally starving, your body starts to use your muscles and break those down for fuel. The body uses the amino acids and protein to form glucose. And proteins are also responsible for tissue repair, muscle building, and immune system support. So you want to be healthy and get the right carbs and the healthy fats to keep your body healthy, to keep your brain healthy, so that you have the proteins that you need to build your muscles, repair tissues, and to make sure that your immune system is working properly. Okay, I want you to try answering these questions and then check in and see how you did. Okay, number 1, what is not a biomolecule? Alkanes. We know lipids are, they're part of the fatty acids. Proteins are, and carbohydrates are. Alkanes are not biomolecules. Now, which of the following best describes a key difference between RNA and DNA? Well, both of them contain thymine, so that's incorrect. And DNA doesn't contain uracil. It contains cytosine. So this one is definitely not correct. RNA is typically double-stranded while DNA is single-stranded. That's incorrect because DNA is double-stranded. RNA is single-stranded. RNA contains ribose sugar while DNA contains deoxyribose sugar. That is correct. And RNA is only found in the nucleus while DNA is found throughout the cell. That's incorrect. That's actually flipped. So, which of the following correctly pairs the DNA bases? Adenine and cytosine, guanine and thymine, cytosine and adenine, and thymine and adenine. So, adenine is never paired with cytosine. It's paired with thymine. Guanine is never paired with thymine. It's paired with cytosine or uracil, depending on whether it's DNA or RNA. Okay. Let's look at these two. What is the correct RNA base pairing for the DNA sequence AAUUUTGGTT? So we're looking at RNA. So we're going to be using uracil. So it's A to T, A to T. Here, I gotta write these down because whenever I look at all these, my mind just starts G to U. My eyes when I see so many letters. So I'm one who has to write things down. And so A, T, T, U would pair with G, G, G. T would pair with A, G would pair with U, U, A, A. I have to write it all down just to make sure. So it's TT. Well, none of these are correct. Because U would go with G, so none of these are correct. But just so you know, it's T, T, U, 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 A, U, U, A, A. So there you go. T, T, U, 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 A, U, U, A, A. Now, what type of bond connects nitrogenous bases in the DNA double helix? Those would be your hydrogen bonds. Those were the only bonds we talked about today. We did talk about peptide bonds, but that was in the formation of proteins. All right, that is it. Let me know if you have questions, and I am happy to help. Study hard.